So today we will start with molecular cloning. Molecular cloning refers to the isolation of a DNA sequence from any species and its insertion or ligation into a vector or plasmid. The vector and plasmid then transform to the host organism. Once isolated, molecular clones can be used to generate many copies of the DNA for the analysis of gene sequence and or to expression of the resulting protein for further study. The clones can also be manipulated and mutated in vitro to alter the expression and the function of the protein. So this is a general diagram of molecular cloning where DNA fragment and vector is treated with specific restriction enzyme and then ligated. The ligating vector then transformed to a suitable host organism. Colonies growing on the plate are selected in respect to the marker present, generally an antibiotic resistance. So what are the steps of molecular cloning? Firstly, the choice of host organism and cloning vector. The selection of cloning vector is important and it is generally depends on the downstream processing of the molecular clones. Then the preparation of vector and the DNA. It can be done through restriction digestions or PCR. Then creation of a recombinant DNA, that is ligating the DNA and the vector. Then introduction of the recombinant DNA into the host organism, that is transformation. Then selecting the organism containing the recombinant DNA, generally with antibiotic resistance. And finally, screening the clones to check the desired DNA insert is present into the molecular clones and also their biological properties. Now we will go through the steps of molecular cloning. So the firstly is the choice of host organism and cloning vector. So for molecular cloning, we need to select the which host organism will be used to replicate the DNA, that is the vector or the plasmid, or the expressing the proteins. For general purpose, bacteria is used frequently due to their inherent property of replicating or making the copies of plasmid. However, bacteria just don't take up the DNA or the vector unless the bacteria is treated and make it ready to take the DNA or the vector. This kind of treated bacteria are called competent cells. Competent cells can be made by two ways, chemically or electrically. For the chemically competent cells are treated with a buffer that contains the calcium chloride and other salts that actually disrupt the cell membrane and creating holes that allow the plasmid to pass into the cell. Chemically competent cells are less expensive, can be made in the lab, and do not require any special equipment. On the other hand, electrocompetent cells requires a high density of cells and a non-ionic buffer. The cells are placed in an electroporation device that delivers a pulse of electricity to disrupt the membranes of the cells. That allows the plasmid to enter the cells. Electrocompetent cells provide highest transformation efficiencies, but do require an electroporation device. The type of competent cells will determine the transformation protocol. That is, chemically competent cells will be used for chemical transformation protocol of plasmid or DNA. So there are a range of cloning vectors are present to be utilized for molecular cloning. Selection of the cloning vector, as we described previously, it depends on the size, cloning method, as well as the purpose of the procedure, that is the downstream analysis. So cloning vectors like plasmid, bacteriophage, plasmid, bacterial artificial chromosome or BAC, yeast artificial chromosome or YAC, or human artificial chromosomes are used. There are some other specialist purpose vectors like the expression vectors or shuttle vectors is very important for biotechnological industry. So plasmids. The plasmids are small circular pieces of DNA that replicate independently from the host chromosomal DNA. That is, it is extra chromosomal. They are mainly found in bacteria, but they are more also exist naturally in eukaryotes like yeast, fungi, or plants. In nature, plasmids provide one of the more functional benefits to the host, such as resistance to antibiotics, degradative function, and or virulence. All natural plasmids contain an origin of replication, which controls the copy number of the plasmid, and typically include a gene that is advantageous for their survival, such as antibiotic resistance gene. In contrast, plasmids utilized in the lab are usually artificial and designed to introduce foreign DNA into another cell. Generally, lab-created plasmids have an origin of replication, selection marker, and cloning sites. The ease of modifying plasmids and the ability of plasmids to self-replicate within a cell make them attractive tools for the life scientist and bioengineer. The table shows the components and their function of a lab-based plasmid. So then bacteriophages. So bacteriophages provided another source of cloning vectors. A phage has a linear DNA molecule. A single break will generate two fragments, which are later joined together with foreign DNA to generate a chimeric phase particle. In genetics, chimera is an organism or a tissue that contains at least two different sets of DNA. This vector is not very suitable for long or bigger fragments of DNA, as long or bigger fragments cannot be accommodated into the phage head.